Okay, all right. So good morning, everybody. Thanks for having me. Thank you very much for the organizers. Let me just see. I can track my time here. Okay, so it's 44. Okay, so I will now turn to a slightly different cognitive function. I'm primarily interested in episodic memory and decisions that we make about memories. So I'm going to talk to you today about how we have used the concept of latent spaces, which is the mission I was given, to understand human cognition, these aspects of human cognition. So what I'm interested in is to understand this really remarkable aspect of our memory system, which is that it is so flexible. You can effortfully search and inquire in your memory to answer questions such as, well, where did I park this morning or yesterday? Or you can do things such as imagine what the face of your mother looks like. How well can you actually do that? And some of you will be able to do that well, and some of you will be not. There's quite a spectrum here. So these aspects of memory, we know well that they rely on temporal-frontal interactions. We know some areas which are critically necessary for this, but the exact mechanisms really remain very little understood. And we want to better understand this, of course, because we would like to know how this system works, but also, very importantly, we would really like to be able to intervene when these mechanisms break down. So perhaps one of the most remarkable aspects to me of human cognition is its flexibility. So I show you this picture on the screen, and I could make you do many different kinds of tasks, some of which I have listed on the slide, for these kinds of pictures. And you could do this effortlessly the first time. You would do 100% correct the very first time you do these kinds of tasks. And these tasks could involve all kinds of cognitive concepts, some of which I have shown here. These are the ones that I have personally studied. So the question is, how do we do this? And what's the right way of thinking about this kind of cognitive flexibility? And there are many different ways of thinking about this, and we don't know which one is the correct one, but two extreme ones would be to think of there's cognitive models, all of which are domain general, and we engage the ones that are needed, a very small subset, and there are many of these models, and most of them will be inactive at any given point of time. So that would be one way of solving this problem. Another extreme way of solving this problem would be to have highly specialized representations. So the substrate that would subserve each of these tasks would be distinct, and when you do a different task, those neurons would do nothing, essentially. So those are two kind of extreme hypotheses. And I'm going to show you now some insight into how we think about this and how we have gained traction in understanding this problem. So now, regardless of which model one subscribes to, this raises a lot of interesting questions, such as how do we switch between different tasks? How you implement this if I give you verbal instructions? How can you do this for a novel task? Yes, you can. So how is that possible? So to gain insight into these questions today, I will show you some data that we acquired in patients who undergo monitoring for their epilepsy, just like Matt talked about. And these patients are being implanted with depth electrodes in various areas of their brain with the purpose of localizing where their seizure is for later either surgical or neuromodulation treatment. And today I'm going to be talking about recordings in two different parts of the brain, the medial frontal cortex, here the DACC and the pre-SMA, and the medial temporal lobe, here indicated the hippocampus as an example. And in these areas we can record a variety of neurons, such as the one shown here, and I'm going to today answer the question of what can we learn about human cognition by looking at a number of these neurons simultaneously and quantifying their population level structure. So one of the questions that I'm going to be talking about is, does the human brain make use of systematic structure in the population? So for example, if I ask you, you have 3 neurons, it's a 3D space, and you would like to configure this space to differentiate 8 different conditions, you could do it in any of these ways I have drawn here and many others, and all of these would work perfectly fine. You could distinguish everything from everything with 100% accuracy, so it would serve its purpose just fine. However, having structure beyond just random configuration, more towards this direction, has certain advantages that you would like to have, such as generalization across details and compositionality. So you could add up things here and be in a different part of the space. So this is theoretically, so the question is this a useful concept to look at human cognition, and how do single cells actually give rise to this phenomena? So I'll be talking to you about 3 different experiments today where we use these concepts. So the first one is I'll be talking about how we use information in memory to make decisions. So over the course of many years, ourselves plus many others in the field have shown that the human hippocampus and the amygdala contain neurons which signal aspects of memory. And there are 2 different sort of extremes of these aspects of memory, one of which is semantic features, such as what we call visually selective cells, that are selective to high-level concepts such as human faces, or monkey faces, cars, or fruits. This neuron here likes human faces. And there is another type of neuron which we call memory selective neurons. These neurons are content-free, but they signal whether the patient has seen that particular picture before or not. And the difference between these 2 trials is single trial. It switches from new to old in a single trial. So we understand this fairly well, these kind of representations, but these representations are not decisions. They are the ground truth, so to say. So the question that I would like to answer now is how can we conceive of this information being used to inform a decision? And for that, I'm going to be talking about data that we recorded in the medial frontal cortex. And the hypothesis here is that the medial frontal cortex receives information from the hippocampus and integrates it to make a decision. And this hypothesis is motivated by a lot of prior work in rodents where there is a monosynaptic pathway from CA1 to MFC, and similar kind of connectivity work in primates. So studying memory-based decision making is very challenging because most often everything is collinear with everything. So to try to tease apart these components, we designed the following task, where we would show patients a sequence of images. For each of them, they are asked to make a decision, answering yes or no, but we would change in a blockwise fashion the instruction. And there were 2 types of blocks, either a memory block where we say, have you seen this image before, yes or no, or a visual question, a categorization question which changed. For example, does this picture show a fruit? So this way we could set up a task where both the visual input is identical and the motor output is identical, but the task that the patient is solving, meaning accessing memory or not, is different. So that's the only difference. So then the hypothesis based on a lot of prior literature is that memory representations in the MTL should generalize between these subtasks, and the subtask here is either memory or categorization, and that this should also be the case in MFC if this is really a domain general decision making mechanism. And you will see this did not turn out to be true. So in the MTL, things are indeed domain general in a very interesting way. So in the MTL, we could train a decoder. This decoder decodes new versus old in one of the tasks, let's say here, in the categorization task, and then train it in a memory task. So that generalizes fine. And the reason why it generalized is shown here. It generalized not because the neural response was the same. The neural response was actually quite different. You see here only 2 of the 4 categories are shown here just for illustration purposes. But you see that what the representation did is that when it shifted from the categorization to the memory task, it shifted in such a way that the configuration between the relative positioning of this 1, 2, 3, 4 dots was consistent. So it was a parallel shift, and that's why this decision plane generalized. So this already answers something about the cognitive flexibility, which is in the categorization task, the new old module, so to speak, is still active, but it's task irrelevant. It's not like that was switched off. That seems to be happening incidentally, even if it's task irrelevant. So now, consider in contrast these neurons from the medial frontal cortex. These neurons behave very differently. So when you look at them, let's see, we look at cell number 2. This cell distinguishes yes from no very well, but only if this decision was based on memory. it does not distinguish yes from no at all in the other task. And similar for here, this neuron differentiates yes from no just fine, but only in one but not the other task. So at the population level of all recorded neurons, remarkably, these representations are very strong. You can decode the patient's decision much better than chance. And this is not shown here, but it's truly a decision. These neurons don't care whether the decision is true or false. But it doesn't generalize. It's completely a chance. There is no generalization. So this model seems to be somewhat specialized to subsets of tasks. And we see this most clearly here, where there's 8 possible different tasks. There were some other manipulations that I didn't talk about, such as the output modality, and there's different also subtasks of categorization. You see here when we look at the one way of visualizing the latent structure of this representation, you see quite remarkably that the first dimension, the one that captured most variance, was not the task that the patient was doing. It was the task itself. Whereas the task that the patient is doing, which is deciding whether the answer is yes or no, is only the difference between these 2 lines and these 2 lines, which is quite remarkable. You see here the block structure shows this very clearly. So this indicates that making memory-based decisions in a way is really special. It seems to engage a different mechanism, and the way that the frontal lobe here separates between these 2 mechanisms is really to map activity into completely different subspaces. So now I'm going to be talking about the second part of our data. And here we extended this kind of thinking to a more high-level cognitive task to see whether different types of cognitive tasks are also special, or whether here perhaps there would be domain channel representations. And you will see here this is a signature of truly a domain channel representation. So one of the very well-known functions of the medial frontal cortex is performance monitoring. So this is this phenomena where you press send in your email and you're like half a second you're like, oh no, I shouldn't have pressed send. That was probably a bad idea. This happens quite often to me. So this kind of immediate reaction. Nobody told you it was wrong what you said. Nobody criticized you. It's purely self-generated. But that's the medial frontal cortex. That's what it does. And this function is very critical in neuropsychiatric disease. It's well understood that it could either be overactive or underactive. So we would like to understand how that works. So the particular task that we use here is the strobe task. So for example, here I ask you to press, what's the color of this word? And you may press green. So then you would say, oh no, that was wrong. So what's interesting in these tasks is that they have these competing demands where we can do this. You may have never done strobe before, but the first time you make an error, you would be able to monitor it. So it has this aspect of being domain general, meaning you can do this across many different tasks. At the same time, to deal with these errors, we require high specialization because, for example, in the strobe task, very specifically, you have to suppress your desire to read. And in other tasks, you have to do many different things. So there are these competing demands. So how is that? How does the medial frontal cortex observe these different tasks so well? So what we did here is we recorded from the same neurons while we asked the patients to do 2 different tasks in different blocks, one of which is the strobe task, and there is another task called the MSIT task, where you have to report as quickly as possible what is the unique number, and there's 1, 2, 3 on the keyboard. And this, for example, would be fairly hard because the answer is 1, but really you press where the 3 key is. So this would generate errors as well. So that remarkably, at the single neuron level, we found many neurons such as these. So these neurons are quite interesting for many ways. First of all, here 0 is the button press. That's when the patient pressed the button. 1 is when the patient got feedback. So you see this signal is fully internally generated. It doesn't depend on external feedback. Second of all, the neuron only responds if the patient make a mistake. It does not respond to any of these different types of conflict. So those of you familiar with conflict monitoring theory, such kind of neuron really is not supposed to exist. And it seems to respond very similarly between the 2 tasks, which is really quite remarkable because the reason you make an error in these 2 tasks is very different. So this turned out to be true also at the population level. So we were able to train a decoder in one of the tasks and test it in the other tasks. And you see it generalizes very well. You can decode whether the patient made an error or not sort of briefly after the button press with very high accuracy and well before the feedback was shown, which is at 1 here. So it's an internally generated signal. And so how does this map onto single neurons? So at the single neuron level, we saw here that really actually most of the neurons turned out to be task invariant. So this is not to say that there are not mixtures, and these mixtures are also very interesting, and then I will show you an example on the next slide of how these mixtures are actually critical. But most of them turned out to be a task invariant. So here we have a clear example of a domain general model that we seem to be able to engage in these two different types of tasks. And we next turn to looking at what the representation of conflict is, of the cognitive conflict. So in the Stroop case, the cognitive conflict is between the reading and the color on the screen. In the MSIT task, there are other types of conflicts. There's a flanker effect, there's a distractor on the side, or there's the Simon effect, which is the button press is incoherent with where the stimulus is on the screen. So first of all, we found this seemingly very puzzling result that decoders generalize between very different types of conflict, such as Stroop or Simon. That seems counterintuitive because they really happen for very different reasons. That doesn't make any sense. Now, it turns out that this is a signature of compositionality. So if we plot, this is just for illustration, the analysis is in the full space, but if we plot the location in state space of where these two different conflicts are related, we can actually see why this happens. And the Stroop here is not drawn, but here you can see that you could easily imagine decoders that either are specialized, they would only detect whether there is flanker or not, or decoders that would just say whether there is conflict or not. The decision plane would be here, and this is the one that's shown here. So it turns out the Stroop decoder will use this decision plane. So this now allows us to test a second prediction, which is, do these neurons retain specialized information, meaning what type of conflict was present? And one prediction of a specialized representation would be a specialized representation that is factorized, I should say, is that it's compositional. So one signature of compositionality is that the vector sum of two individual components should land where the neural state would be if both are present. So here this was very much the case. So the theoretical prediction is indicated here in dashed, and this where the data actually landed. So you see here that this kind of representation that I illustrated in the beginning is really actually formed here. And you see that this is a parallelogram, but not exactly. It's slightly distorted. And these distortions turn out to be really critical, because if the parallelogram would be perfect, you couldn't solve certain problems, such as the XOR problem. And it turns out that these distortions are significant. They are related to these task-specific neurons, these 17% on the prior slide. So one example of this is shown here, which is that if there's a nonlinear interaction term between the task and some cognitive variable of a given neuron, that will predict this distortion of the parallelogram, so the deviation from the prediction of the linear sum. And so this turns out to be a critical aspect. So here we have now shown that at least in this performance monitoring domain, it is possible to represent neural states in such a way that you can both generalize and you can still retain specialized information by forming these distorted parallelograms. So lastly, I'll talk about a new experiment that we are still working on, and this experiment will answer the question whether the kind of latent representations or these low-dimensional spaces that I have shown now can also be learned. So far they were all there by instruction. They're just the patient already knows this is a relevant feature. They're like for conflict, I'm telling them this is the correct answer. So the question is, can these be learned? And if so, how are those relevant for learning? So to address this question, we performed a new experiment which superficially seems very similar, but which has a latent structure. So we show the patients, again, a series of pictures. Here there are only 4 possible pictures. And they have to learn a stimulus-response mapping. So for example, here, the correct answer is left. For this picture, the correct answer is right, and so on. And they will do this for a certain number of trials, and they will get feedback whether they're correct or not. And after some period of time, there will be a switch to a different task, but here the switch is uncued. They don't know this. They have to infer this from the feedback. And the switch is systematic in such that the response mappings, they invert, and they are also coupled. So for example, if the patient understands that the response to A and C is the same, if they get feedback, an error feedback on only one trial, they should theoretically be able to infer the correct responses to all the other trials. So if they can per inference, and if they properly represent this latent contextual variable. So remarkably, subjects can do this quite well. So this is shown here in that it takes some time for patients to acquire this skill, either by practice or by verbal instruction. I'll get to that in a second. But once subjects have learned to perform this behavior, those are the blue lines here, you see here that the first time they see a given stimulus, such as, for example, A, after they got an error, so here they see the B stimulus, they don't know there was a switch, so of course they're significantly below chance. The first time they see the A stimulus, they perform successful inference, and this is for the other 2 stimuli. So now we asked, what's the difference in the representation of the underlying contextual variable between the sessions or the trials where the patient doesn't have inference behavior, so they have to relearn from scratch, versus where the patient has inference behavior. So I'm going to be using 3 tools to quantify the structure of the population, and I'm going to be showing you data from the hippocampus, which turns out to have a very privileged role here in that it's very distinctly the first area that shows this difference. So we are looking at different dichotomies, and the most important dichotomy that I want to be focusing on here today is context. So there are the 4 stimuli and they can appear in 2 different contexts. So can we differentiate between the 2 contexts, which were never cued, and the subject was not told that there's these 2 different contexts. And it turns out that once the subject is able to perform inference, this contextual variable becomes present. It emerges in the hippocampus. Not only does it emerge, but it's in a very specific format, which we call abstract. It's in an abstract format because, first of all, there is cross-condition generalization, meaning I can learn the contextual distinction between some of these pairs and it will generalize to other pairs. And also not only is there generalization, but these coding vectors really are parallel. So as the subject learns to understand that there is a contextual variable, these coding vectors become more parallel. So does this matter behaviorally, or is this just some kind of a thing that emerges alongside this learning? It turns out we have 2 pieces of evidence that show us that this kind of representation is behaviorally relevant. One is that when we take the blocks, on average, we know they do well, they do inference, but they still make errors once in a while. When we look at those blocks, in the trials where they do correct, the contextual variable is present only when they do correct, but it's not present when they make an error. And I'm showing here only the decoding, but the same thing is true for the other 2 metrics, the CCGP and the PS metric. Second of all, there is this interesting situation that some patients will learn this task the first time they perform it. They will just figure it out and they will have a moment. They're like, oh, okay, I get it now. But others don't. There are a chance. So then we take the opportunity to just sit down with them and tell them, look, this is how this works. There's a contextual variable, it switches and so on. The things are tied together as the table. We'll just show them the table. And then something really remarkable happens. Some subset of the patients then say, oh, okay, I understand now, and then they do it. And so if they then can do it after we gave them verbal instructions, right away this kind of representation, here we go, emerges. So an abstract representation emerges, and the patients that then still can't do it, the representation has not emerged. So this shows us that this kind of abstract representation at the population level really does give us information that goes beyond of what we could glean at the single neuron level. And so why is this? So we try to gain insight into what exactly happened here, and I'm just going to summarize very many kinds of analyses using this sketch here, which he borrowed from Stefano, which he kindly made, which is what we find here is that several things happen. First of all, the distance between the different variables increases. So here, the distance between the different, so here we just show free stimuli for visualization. The distance between that's enforced by the context increases. Now, something else happens as well is that the variance, which I've just illustrated here using this balloon, around each of the condition reduces. That we also found. So those you could all do using single neuron tuning changes. But in addition, what also happens is that it's not just that this thing is just expanding and the variance goes down, but things actually straighten. So this is what we find. This is the parallelism score that I showed earlier. That things become more straight and more parallel. And this happens here as a function of learning, either due to verbal instruction or due to practice figuring it out. So to summarize, I have shown you here that we can use geometric tools to systematically investigate the structure of cognitive representations, and that this structure is predictive of different kinds of behavior. There are indeed category familiarity and latent context in the hippocampus, which are domain general. So there's some signature of the original model that I sketched out. This is also true in the medial frontal cortex for error and conflict. However, many others this is not always the case. So choices, most importantly, are not. So choices don't seem to be domain general, at least across the categorization and memory boundary. And I've shown you how we think about how the same group of neurons can represent both domain general processes as well as generalization by being structured in a way such as illustrated here, which means that if you're a downstream readout listening to these neurons, all you have to do is change your weights to either generalize or specialize, which is very different from a typical kind of low-dimensional representation, which we think of as compression. Basically, you lose information by building that low-dimensional manifold. Here, that really wasn't the case. It's just restructuring the information, but it's not eliminating it. It's not compression. It's a restructuring. And so overall, this illustrates that aspects of executive function, which is here I use cognitive flexibility, can be understood as geometric transformations in neural state space. So with that, I will conclude, and I'll thank the many individuals involved in this work. In particular, I've highlighted here in bold the students and postdocs who have directly contributed to the data I have shown, and Christos is actually over there in the audience. He participated in the last project. So thank you very much.